And I think the last talk of the session is from Courtney Sakolish. All right, from TexVal. So thank you.
Last but not least. So yeah, thank you for having me. Usually it's Yvonne up here giving this talk, but he sent me up this time, so we get a little bit of a different perspective on the consortia. So I'm glad we talked about space earlier because I'm about to give you guys a space level analysis of what we do at the consortia and how we try to build credibility and confidence in these models. Okay, and I'm going to try to figure out how to use the pointer because it's been fighting back with other people. Awesome. So yeah, the consortia I feel like is a really great example of kind of trying to onboard these and test these models before they're brought on by the end user. So there's kind of been the idea of building these really cool complex models, publishing on them, but we're not really seeing them being adopted for decision making context. So again, it's about building confidence and building credibility in what these models are actually about and how they can be used within each of the organizations. So we started back in 2016. That's when I joined the lab at Texas A&M on this tissue chip testing center. So we were one of the two testing centers that were funded as part of this NIH NCATS group. And again, you'll see a lot of really familiar names and faces in the audience today. So again, thank you to everybody that we had worked with. This was a really great experience working with around 20 different models, and it was a really big learning experience, I think, for both sides. This kind of gave us an idea of how we evaluate models as part of the consortium. So it's based off of sort of a 4-tier approach. And I'll focus kind of on tier 1. I think this was the main goal of the testing centers was reproducibility testing to build confidence. So this was replicating key published findings by the developers, and this was establishing these really robust protocols and getting these systems working in the lab. And then once we had a good idea of the types of data, the throughput of these systems, we could move then on to tier 2, which is sort of defining the context of use and how these could actually be applied. So again, there was a lot of learning that happened during this 4-year period. Did we get the developer lab made devices to work outside of the lab, and were they reproducible? I think for the most part, yes. And that's, again, kudos to the developers for making these robust systems that we could actually work with. I think the biggest challenge to tech transfer, and this is a universal issue, and I'm not going to get into details because we could have a whole talk on this, but it's the availability of reproducible biology. To have a reproducible model that you have confidence in, you need to be able to have the same source of cells that you can carry over these longitudinal studies and compare across different platforms. So that's kind of been a continuing issue. And then I think the most important learning from the study is obviously we learned a lot about the operation and phenotyping of these models, but it also gave us an opportunity to work with the developers and kind of give them an idea of the pressure points of their systems and help them to refine these models to be more useful to the end users. So after that initial 4 years, we moved to a consortium model. NIH had pushed the testing centers to have revenue generating activities beyond the initial funding. So Yvonne and I started to knock on doors, make house calls, and bring together people that we thought would be interested in actually using these models and who wanted to maybe product test them and learn more about them before actually onboarding them. So again, we have a diverse membership from industry, trade associations, government agencies who can define a common work plan. Surprisingly, there's a lot of crossover in interests of what each of the members are looking for in terms of tissue chips, chemicals, phenotypes, platforms, et cetera. And then at A&M, what we do at the consortia is to actually execute on that work plan. So we just had our last planning meeting with the consortia about a week and a half ago, identifying these chips of interest, these types of phenotypes that we'd be looking into. But again, procuring equipment, learning about the true cost of operating the systems, establishing the models in the lab. And again, this goes back to identifying the appropriate biology to put on these systems as well. And then replicating key published findings, but I think importantly, trying to refine the models based off of feedback from the consortia as we're kind of getting data and experience in using these systems. So is there value? What's the value of the consortia? Basically, we ask for a $100,000 a year membership fee from each of the members, and this all goes towards research. Each membership provides 2 to 3 scientists to participate in meetings. Again, we don't want to have too many cooks in the kitchen. I think this is a good number of people who can actually meaningfully contribute to assembling the work plan and refining these models. And again, they have input on what we're actually testing each year. So that's what you're getting out of the consortia. But also you're getting access to all of the data, the protocols, everything that's generated within that year period. You have basically unlimited technical and scientific support from myself and from other scientists in the lab who've worked with many, many models now at this point. We publish extensively. I'll show you on one of the later slides, doing these cross-model comparisons, that's really important. But I think lastly, and maybe most importantly, is the opportunity to engage in these really open discussions with other members of the consortia. People are very forthcoming on what's working in their lab, what's not working, what they've tried, what they want to see. And I think being able to share and learn from each other in this kind of context is really important. So who's in the consortia? Well, this is the last, I guess, 7-ish years of membership now. So we started in 2020 and we're up to 2026 now. We started with 5 members and then we're slowly kind of increasing. And if anybody wants to join the consortia, we're still looking for more members. So talk to me or Yvonne after. You'll see the tall person kind of hovering above the rest of the crowd. That's Yvonne. But yeah, so we have a good, I think, diverse membership, as you can see. And most importantly, we're getting renewals every year. So people have some perceived value of this kind of membership, this kind of organization. And I feel like we're getting valuable data from this. So again, the beginning of the year, we ask each member, what organs are of interest, which tissues are of interest. I'm not going to go into the specifics of the models or the phenotyping, but I'll kind of just hover on the tissues here for a moment. And liver, not surprisingly, has consistently kind of been at the top. So in 2026, this year, we've had 7 of the 9 members that asked for liver. And then blood-brain barrier also kind of popped up this year. So it's kind of cool to see these trends of what people are actually interested in using within their companies. So I'll focus a little bit on liver and then give you an example of some of the kind of cost comparison analysis we've done on BBB in just a minute. But again, as our consortia is growing, we're starting to get more interest in different platforms. So we're trying to keep a pretty tight-knit group. But again, we are looking for new members if you're interested in joining. The colors shown in green, these are the platforms that we actually tested within that year, or in the case of 2026, we're hoping to test. So we are getting to a lot of different platforms. Again, back on that value proposition. So for a measly $100,000, we're testing thousands and thousands of chips, a lot of different organ systems, and this is not something that any member of the consortia could do individually. So I think having this shared cost sharing, this shared de-risking of these platforms is beneficial in that way as well, that we can test so many different conditions, so many different chips. And again, we're really focusing on commercially available models. So you'll see up there, we're testing what is operationally ready for actual testing and scalability. You'll also notice that the number of conditions and chips is increasing each year as well. That's on the bottom. And it does feel like we're working harder and harder every year, and we're having the students work harder. But I think the main trend is that we're moving away from the more complex models, and we're moving into some simpler models like spheroids, like plate-based models. And that's really been kind of the interest of the consortia, is moving towards these more scalable models and higher throughput systems. So again, building consensus, building confidence is about having evidence-based qualification. So we follow 4 main pillars for qualifying each of the models that we test. The first one, and I think this is the most important, and Kim kind of alluded to this earlier, but having comparative analysis of models and cell types and sources. So we're not just looking at, one liver chip. We're looking at, any liver chip that we can get our hands on for the most part, and looking at how those chips stack up against one another, how they stack up against 2D, against spheroids, and doing these comparisons and being very transparent about how these models stack up against one another. And then also looking at cell sourcing, so from different donors, from different individuals. Again, just having these really transparent comparative analyses, I feel like, is very important. We also do comprehensive but sensible phenotyping for each experiment. So having imaging, biochemical data, analytical, and then model omics. And again, Kim did a great job explaining what model omics is, but it's looking at the basal and treatment effects across these different platforms, looking at differences in sensitivity and specificity of these models. Cost benefit analysis. So again, she showed she did a great job introducing my talk, I think, but the rat where you have all of these different tissues versus the single chip that's maybe $500. So it's not just about the consumables, it's also about purchasing that equipment or leasing that equipment, and the cost to operate. So how many, man hours or woman hours does it take to actually operate this system every day, to phenotype these models? What are the consumables costs? How much does it cost to actually run an experiment? What's the failure rate? Because again, if you have a more complex model, you're going to have issues like bubbles, tubing. There's other kind of pressure points that you might run into. So you may need to increase your number of replicates to get the same kind of data. And then also, what other types of instruments are needed? Do you need specialized equipment? Can you just use kind of general lab equipment to test these models? And then lastly, keeping the domain of applicability broad. So we're not just looking at one class of compounds, we're looking at drugs, chemicals, environmental compounds, and looking at different exposure modalities, different mechanisms for toxicity, for metabolism, and just trying to have a fair comparison across all of the different platforms that we're looking at. So I'll kind of talk a little bit about some of the comparative analysis we've done. And like I said, liver's been a big focus, not just for the consortia, but also for developers as well. There's a lot of interest in the liver space. And each developer has their own kind of ideas about having multi-cell, having flow, different sorts of aspects they're trying to achieve. So within the first few years, kind of coming out of the TCTC Center and leading into the TexVal, again, we're really focused on platforms that are commercially available, things that are scalable that we can really purchase and do large-scale experiments and do these longitudinal studies off of. So you can see here we've done a number of studies on the LAMP model as well as CN Bio, Nemetis, and we did a number of publications on these systems looking at those comparative studies. And again, publications are defined by if that chip is available and if we can actually get it in-house and test this. So more recently, we are still working with those systems. Again, we have new questions the consortia is asking. We always go back to those other systems because we do have them in-house. We have a lot of experience on them, but we're adding new things to the mix as well. So we've onboarded the tissues system, as well as micropatterned co-cultures or MPCCs in collaboration with Salman Katani's lab. We've participated unofficially in the tissues liver ring trial. So I actually went and trained on that system with the other members. We got the same batch of cells, the same lot of hepatocytes, same compounds, and we followed the exact protocol of the ring trial, and then we're able to share that data back with them. So this is really interesting. And again, we're not trying just to do the experiments within TexVal. Whenever we can do these collaborative cross-laboratory comparisons, that's very important in terms of looking at the reproducibility and the confidence in these models. We've also been doing a lot of comparisons now with spheroids, so static versus fluidic spheroids, in addition, of course, to 2D and a lot of the MPS systems we're working with. Looking at different species, we've done a lot of cross-model comparison, looking at cross-species versus human, also looking at primary versus HEPA-RG versus IPS. And then we've been kind of dipping our toes into some gut-liver co-culture multi-organ models using the tissues and the transwell-based models as well, and then trying to go back and do that in some of the other systems that we have in-house. And of course, always looking for new models that are coming out. So again, if you're a developer that has liver chips and wants to work with us and is willing to lease your system, please let us know. Just really quick, an example of a cost-benefit analysis. So like I said, there's a lot of interest this year in BBB. So we're bringing this model back. But in the previous years, we had established our own kind of in-house BMEC model based off of the Schuster protocol, the paper shown on the left. And this is, old protocol, but it works very well at establishing these really nice, tight brain microvascular endothelial cells. So again, trying these in all different conditions, trying to figure out what the most optimal condition is and have a reasonable cost per sample. So growing them in 24-well transwells and 96-well transwells. In the CN Bio, do you need flow, having different flow rates, seeding these with and without astrocytes and pericytes from primary donors, and then compared this against a commercially available model, which is the CDI BBB model. And you can see here that while we got all of these systems to work in the lab, there's quite a difference in terms of that final column there, the cost per sample. So, and again, the throughput of the systems as well. If we're doing these complex models in, with fluidic shear stress, that also reduces throughput. So depending on the questions that we're asking, you may not need the most complex, most expensive model. You might be able to get away with a simpler model in that case. So we do publish regularly. I have just 2 examples here. I'm going to kind of move through this fast because I want to have time for questions at the end on the consortium. But we do a lot of these kind of as Kim said, non-sexy analyses, but I feel like transparency can be sexy in its own way. But so again, the purpose of these papers isn't to say which is the best model. It's to just put the information on there about what the limitations of that system are, what the benefits of each of these models are, and then to let the end users be able to decide how they want to use these models and how they want to use that information. So we do publish regularly on this, and you can see that in the last few years, we've published over 20 papers doing these comparative analyses in addition to the other research that's going on in the lab. But this is really important. And for all of these publications, we put the data in the EVE Analytics database and make sure that this is publicly available. So again, we're trying to be as transparent as possible so people can make decisions about which models they want to onboard. So this is my last slide. So is the consortium a success? I might be a little biased, but I would like to think that it is. So we were able to bring together a collaboration of diverse stakeholders. And again, they're renewing each year, and we're still looking for more members. If you'd like to join, talk to us. But I think the main value proposition is try before you buy. So you're not locked into a specific system. Once you buy that system, you're kind of keyholing yourself into using that, but this gives you an opportunity to test and product test that system and learn about some of the kinks and some of the drawbacks in a realistic setting before you actually onboard that. And there's a lot of learnings of the consortia, as I had mentioned before. And not all that glitters is gold, but we're trying to be as realistic and really push these systems as much as possible because, it's not the goal of the consortia to make every single chip work, but it's my own kind of personal goal as an engineer to make everything work in the lab. But I think the main takeaway, the main success of the consortia is, not always the decision to onboard the model, but also it can be the decision not to onboard the model. It's, again, just providing that information and being fully transparent about these systems and what they can and can't do so that the end users can make their own decisions on whether to onboard. So just to conclude, I think, the main thing to build credibility is transparency and having that sharing of data so that everybody has the same kind of groundwork and then can decide, how they want to onboard and use the system for decision making. So thank you. I'll take any questions. Or not.
Keyword for most of your talk was really transparency. And do you think that some of your ability to provide that comes from the fact that your funding isn't tied to the success of a device?
Yeah, I mean,
pretty much everyone else's.
Yeah, no, absolutely, right? So we're buying. Oh, sure. The question was, do we think that our data is more transparent because we're not tied to a particular company, right?
No, it's about the fact that your funding.
The funding.
That pays your salary is not tied to the success of a platform. The platforms can all fail. They can be absolutely all trash, and you still have a paycheck at the end of the day.
Yeah, I feel like that does help, right? We're not trying to make every system work. We're trying to give a fair comparison across platforms, and I think doing those cross-platform analyses, that's important, and also working with the developers. So we're not trying to fail. It might seem like we're trying to fail every model, trust me.
No.
But we are trying to make them work. We're providing information back to the developers as well, the companies that we buy these systems from, and trying to help them make these systems more robust and help them kind of do that. So yeah, I do think that's helpful. And again, as you said, our success is not having the chip onboarded. It's just providing information so that the end users can onboard them. We're not onboarding any of these ourselves. We're just purely evaluating them against each other,
Okay.
like the Thunderdome of chips.